Hi, it's Dr. Ogden. We're going to be looking at DNA, its structure and function, and some other related issues to DNA. It took a number of years before scientists recognized that the molecule that carried the information, the genetic information, was DNA. But once that was determined, basically there were a number of different labs that were trying to find out what was the structure of DNA and how is it that it carried the genetic information. And one of the most important players in this process was a woman by the name of Rosalind Franklin. And she was this amazing scientist who spent lots of years and became very good at doing what's called X-ray crystallography. So this is where you take an X-ray source and you shine it through this LED screen. It then becomes a beam of X-rays, which then go through the sample of whatever it is you're trying to determine its molecular structure. And so in this case, DNA. And so you had to crystallize DNA, and it would then have the X-rays shine through it, and then you would get this diffraction pattern that would be able to be detected by this plate, and essentially you would make kind of a photograph, right? A black and white photograph of the pattern of the spread of the X-rays as they would come through the crystal. And so she made lots of these different pictures, and a series of these became the ones that were very important to leading to the discovery of DNA. And this is one of the most important ones right here, this photograph. So she actually never found out the exact structure of DNA, even though she was working on these photographs. It turns out that two other men who happened to be in the right time looking at the right things and then putting all the pieces together actually came up with the structure of DNA. And this was Watson and Crick. So they're the ones that wrote a paper, and this is the very first line of their paper. I love this. It says, "We wish to suggest a structure for the salt of deoxyribose nucleic acid, DNA. This structure has novel features which are of considerable biological interest." I always think that's probably the understatement of all science sentences ever, because really DNA has been the key to modern biology. And Watson and Crick were able to actually see those photographs that Rosalind Franklin had made, and together with the models that they had been working on, they were more theorists, they were able to put it all together and come up with the structure of DNA. And so we now know that DNA is kind of like a ladder that's twisted on itself. So it's a double helical twisting molecule. And we've looked at this before, but if you look at the side of one of the ladders, then it's made up of three parts: a sugar, a phosphate, and a nitrogenous base, as seen as over here, right? The sugar, phosphate, and nitrogenous base. And those bases can either be A, G, C, or T. And then the sugar and phosphates alternate and become the backbone, with the nitrogenous bases sticking off to the side. And then you put those together and you get the double-stranded DNA molecule. And G's always bind with C's and T's always bind with A's. And these are just three ways to represent the DNA molecule. So something that's important about DNA is because it carries all of the genetic information, it then must be carried throughout each successive generation. So when does replication of the DNA occur? We learned this before, and this was in the synthesis phase, right prior to mitosis or meiosis, in the S phase, during interphase. And so DNA replication was also discovered to have a really interesting way in which it's carried out. It turns out that DNA replicates itself very similar for bacteria or for eukaryotes. In bacteria, there's one starting point, and it goes all the way around the circular piece of DNA and then finishes. In eukaryotes, because of the size of our genomes and we have these linear chromosomes, you have multiple origins of replication along a chromosome. So there could be one chromosome with multiple origins of replication. And then you get these bubbles that form, and the DNA is replicated, so copied, right, until the bubbles run into each other. So it's as if you start with the parental strand, you unzip it, and this is what Watson and Crick even postulated was possible, is you could unzip DNA and use each side as a template strand to then form a new strand on the other side. And what you end up with then are two daughter DNA molecules that are copies of each other. And in genetics, remember, we refer to these two daughter DNA molecules as sister chromatids. So this is what happens across eukaryotes when they go through DNA replication with these multiple origins of replication until you end up with the two daughter DNA molecules. Now, I want to just make mention of one molecule in particular that's really important in this process. There's a multitude of different proteins and enzymes that are involved in this, but one in particular is called DNA polymerase. And DNA polymerase is the molecule that does the copying. And it's a very faithful molecule. It makes very few mistakes, but it does make mistakes every once in a while, and of course we call those mutations.